Hello guys. Today I will discuss something on we call it a molecular cloning. So, so this molecular cloning basically deals with uh, a particular set of DNA that you want to clone in a particular organism. So to execute that same thing, we need a cloning vector. So what does a cloning vector mean? And cloning vector means that it should have certain properties, like it should have one multiple cloning sites where there are some the places where different restriction endonuclease can basically cut. A particular cloning vector should have a origin of replication. So once the cloning vector is being incorporated in a particular microorganism, so it can has a replicative property. Third one cloning vector should have one selectable marker gene so that we can select a transform cell with the help of certain selectable marker gene, maybe an antibiotic resistant gene like kanamycin or like ampicillin genes are very responsible for it. Now to execute the same thing, suppose we have a plasmid vector. So what we need to do, the gene of interest that we want to clone, so we have to code both the gene as well as the plasmid multiple cloning site with the same restriction enzyme, so that in the plasmid multiple cloning site, or we call it a MCS, that should have a specific restriction cut that should have the same restriction cut of the both side of the gene that we want to clone. So once both the gene as well as the plasmid MCS site are basically cut with the same restriction enzyme, so what we can do, if we keep these two and incubate in a microcentrifuge tube with the enzyme called the DNA ligase, so what it basically does, so it basically this gene of interest can be ligated in that plasmid to form a recombinant plasmid. Now this recombinant plasmid can be incorporated within the bacterial cell. The process is called basically bacterial transformation. So once bacteria is basically transformed, we can grow the bacteria, and as the bacteria will keep on reproducing, we'll be getting lot and lot of bacteria that harbor that plasmid. Now the question is that how to select that particular bacteria which harbors the plasmid, so compared to that those bacteria which does not have which do not harbor the plasmid. So here comes the use of this antibiotic resistant selectable marker gene. So once we grow the bacteria in a suitable media like a luria broth or nutrient broth like this, so what we can do, if we make a solid agar media containing that antibiotic, maybe ampicillin or kanamycin, so what happens, once we try to follow a simple plating technique and we incubate the plate overnight, so only those bacteria will grow, those harbor only that plasmid containing that drug resistant gene, and those bacteria which during the transformation which do not contain that plasmid, the plasmid that contains the antibiotic resistant gene, that will not basically grow. So here is basically the how do you basically select this. So to make you better understand, so I am going to share with my screen with a very informative animated video. So I think once you go through the video, so you will better understand the same thing. So just wait, I'm going to share. So I think you all will enjoy. So just wait for a minute. So I'm going to share this. Now you enjoy, please.
Molecular cloning is a process of isolation of a specific DNA fragment and transfer of this fragment into a plasmid vector.
Exactly.
As a part of a plasmid vector, the DNA fragment could be easily amplified, sequenced, stored for long periods of time, and used for gene expression and other functional studies. Two starting ingredients of molecular cloning are a plasmid vector and a DNA fragment that has to be inserted in it. The DNA fragment is usually a gene or other functional
So two things that we need. One is we call it a vector, that means the plasmid. Another one is the insert, that means my gene of interest. So these two things that has to be cut, and simultaneously that has to be joined to form a recombinant plasmid. And this recombinant plasmid, with the help of the process called transformation, that should be incorporated within the bacterial cell, and we can select with the help of selectable marker gene. Now again I am continuing.
A region from a living cell, or it could be an artificial sequence with properties useful for a researcher. Plasmid vector is a circular piece of DNA that could be easily amplified in E. coli, stored for long periods of time, and easily manipulated in a test tube. A typical plasmid vector contains an origin of replication that allows it to be replicated inside a bacterial cell, a selection marker, for example, a beta-lactamase gene coding for ampicillin resistance, and a multiple cloning site, which could be cleaved with several restriction enzymes such as BAMH1, ECAR1, or NCL1. In many vectors, the multiple cloning site is surrounded by sequences of promoter and terminator that guide expression of inserted genes after the vector is introduced inside a cell. To be used for molecular cloning, both vector and insert DNA are treated with restriction enzymes that cleave double-stranded DNA molecules, producing overhanging single-stranded nucleotide tails.
So here we have taken two different restriction enzyme. One is NCO1, another one is a BAMH1. So we are going to use these two restriction enzyme both to cut the plasmid vector as well as the insert.
After their ends have been prepared with restriction enzymes, vector and insert are combined together, and another enzyme called DNA ligase is added to the mix.
But one thing I need to mention, that each and everything that you are going to take with your using your micropipette, so always remember that you should not introduce any cross contamination. That means the micropipette tips you are going to use for taking the vector, that should be changed before the micropipette is used to take the insert. So now the micropipette tips cannot be used for each and every sample that you are going to take. So you should pick one pipette tip for taking the insert, one when you are going to take it the vector. So that means each and every time you have to make yourself more sure that you are not going to introduce any cross contamination, right?
And while complementary base pairing of single-stranded overhangs brings the ends of vector and insert together, the DNA ligase fuses them into one intact DNA molecule. In order to make multiple copies of this molecule, the ligation mix is introduced inside the E. coli cells in a process called transformation. During the transformation, the cell DNA mixture is kept on ice and then exposed to 42 degrees Celsius. A sudden change in temperature drives the DNA inside some of the E. coli cells. Then the cells are plated on a plate with growth medium, supplemented with a selective antibiotic.
So before the specific transformation, so what we need to do, we have to make the bacterial cell competent. So ice cold calcium chloride is basically used to make the bacterial cell competent. So how to make the competency, that I will describe in another section. So today just I am going to describe that how molecular cloning is basically executed. So what is the use of the cloning vector? So how a recombinant vector is used by putting the insert here, and how the process called transformation that is being performed to put the recombinant vector inside the bacterial cell, and once how to select the bacteria with the help of the antibiotic resistant marker gene.
Only the cells that acquired the plasmid have resistance to the antibiotic and can grow on such a medium. After overnight incubation at 37 degrees, each transformed cell produces a colony of identical cells, oftentimes called a clone. The selected clones are then individually picked, grown even further in a liquid medium, and the DNA is extracted from them. So, in the process of molecular cloning, a DNA fragment that represented a tiny fraction of cell genome is integrated into a bacterial plasmid. As a part of the plasmid, this DNA fragment represents a quarter or more of total DNA in a test tube, and it could be effortlessly and endlessly amplified in E. coli. This presentation was brought.
So anyway, today we describe that how a molecular cloning technique is basically utilized in laboratory just to make several copies of your gene of interest using a suitable cloning vector and a process called transformation. Anyway, in the next class that I am going to describe something on how to make the cells competent and how a genome sequencing is basically done after a suitable condition, and we will again explain something on how PCR is basically done using a suitable primer. Okay, bye bye.